So as you can see that our today's topic is oogenesis. Oogenesis is formation of mature female gametes. The process of formation of mature female gametes, that is ova formation, that is called as oogenesis. Oogenesis is a very complicated process, extremely complex process. It starts in a female even before her birth. Even before she is born, in her fetal stage ovaries, oogenesis begins over there. And it continues even after puberty till the menopause sets in. The process continues over there. This makes the process extremely complicated one. For our convenience in a study, we are dividing the process into 3 stages: events related with the oogenesis prior to birth, called as embryonic stages. Then, second stage is oogenesis related events between birth and puberty. That's a second stage. And lastly, after the puberty. So, third stage is referring to oogenesis events related after the puberty. So, this is how we divide our entire oogenesis topic into 3 stages. To start with the first one, that is embryonic stages oogenesis activity. If you can recall the structure of the ovary, the outermost covering or outermost layer over there is what is called as germinal epithelium. That is termed as germinal epithelium. Immediately then after, there is tunica albuginea. And then there is a remaining part, that is the stroma portion of it. This is the germinal epithelium. This is the tunica albuginea. And then there is a stroma region. The cells of germinal epithelium, the cells of germinal epithelium. In the fetal ovary, when the female is not even born, she herself is a fetus in the mother's womb. At that stage, in her ovary, these germinal epithelium cells at regular interval undergo repetitive cell division, mitotic division, and they make clusters of diploid oogonial cells. So oogonia cell masses are created. You can see that like this, the cell masses are created. These are the oogonial cell masses. So such oogonial cell masses are created in the embryonic stages of the ovary. How many such cell masses? Millions in each ovary. Approximately million plus such oogonial masses are created. Diploid oogonial cell masses, or you may call it as the gamete mother cells, their masses are created with the number like millions or more than that. In each cell mass, centrally placed cell, one of the oogonial cell, and that one which is in the center normally, it differentiates and transforms itself into what is called as primary oocyte. it is transforming itself into what is called as a diploid cell named as primary oocyte. Each mass of oogonia cells in the center develops one of these cell into primary oocyte. Obviously, this is comparatively little larger than the rest of the oogonial cells in the given mass. However, all of them, the surrounding oogonial cells and the centrally located primary oocyte, all are diploid in nature. These primary oocyte starts meiosis 1, and in meiosis 1 there is a prophase 1 stage wherein it gets arrested. So almost as if starting the meiosis, particularly the prophase 1, and before the prophase 1 is getting over itself, it is being arrested. Leptotin, zygotin, pakitin, diplotin, and then cell is arrested. So it doesn't go in a dikinesis and doesn't complete the prophase 1. So the primary oocyte is the only one in this cell mass that is undergoing meiosis. It just starts meiosis and arrests itself in the meiosis prophase 1. So that is the real primary oocyte surrounded by these oogonial cells. These are obviously oogonial cells in its surrounding. Together, these two make what is called as primary follicle. Now, this is not just a simple ordinary oogonial cell mass formed initially by the germinal epithelium. Millions such cell masses in each one, centrally located oogonial cell transforms, differentiates into primary oocyte, comparatively larger, otherwise it is also diploid just like other oogonial cells. And this primary oocyte starts meiosis and soon it arrests itself in prophase 1 part of the meiosis 1. And now onwards, it is called as primary oocyte surrounded by this oogonial cell mass is now known as primary follicle. Such million primary follicles are formed inside each fetal ovary. Each fetal ovary prepares millions such primary ovarian follicles. With that, the first embryonic stages of the oogenesis is over. Second is about just a minute. Yes, here you can see that. Have a look. The germinal epithelial cells, they undergo continuous mitotic division and make a cell mass. Amongst the cell mass, oogonial cell mass, diploid cells. One of the cell is differentiating itself into primary oocyte. These remain surrounded by the other cells, which are oogonial cells. And together it is called as ovarian follicle. Now in this ovarian follicle, the primary oocyte will start the meiosis 1, first meiotic division, and soon gets arrested. At this stage, the ovarian follicle, it is termed as primary follicle. It is termed as primary follicle over there. such million primary follicles to be found in each ovary, fetal ovary, I would say. And then the birth is given to the female, to the baby. In that female, since birth till she reaches to the puberty, in the second stage of the oogenesis, large number of primary follicles will degenerate. They were there na? In each ovary, nearly million such primary follicles, each with a centrally placed larger diploid primary oocyte, which has been arrested in its ongoing meiosis at the prophase 1 stage, surrounded by layers of uh, Ugonia cells. These Ugonia cells are called as actually granulosa cells. So they are termed as granulosa cells. So the primary oocyte and surrounding granulosa cells together form what is called as primary follicle. Now, after the birth, till the puberty, 12 to 13 years age, in the body of a female, in her young ovaries, these primary follicles undergo degeneration. How many are left there? So in each ovary, approximately 60,000 to 80,000 are left. When you said that when we said that initially there were, 1 million ovarian follicles, primary follicles in each fetal ovary. By the time of puberty, out of that 1 million, how many are left? Almost 80,000, which is 8% of that 1 million. So 95%, 92% of these primary follicles actually undergo degeneration. Hardly 60,000 to 80,000 are left in each ovary as the primary follicles. These follicles will appear like this. You can see in the pic. These all are primary follicles. Now the oogenesis enters into the third stage, which is called as puberty. Since puberty, 12 years plus age, female starts experiencing 2 cycles in her body. Related to her uterus, she experiences menstrual cycle. And related to her ovaries, she experiences ovarian cycle, which is nothing but the part of oogenesis process. These 2 cycles go for 28 days and they go parallel. The day 1 of ovarian cycle is actually the day 1 of menstruation and menstrual cycle. So since the day the menstruation starts in a female in the given cycle, that is the first day of menstrual cycle, and actually that is the first day of ovarian cycle also. So these 2 cycles run parallelly in her body. So in these first few days of the cycle, now when I say cycle, you have to understand menstrual and ovarian cycle both, and because they run in this parallel manner, so each one starts parallel to the other. So if day 1 the menstruation starts, because it is apparent phenomenon, we can make it out. So since that day onwards the menstrual cycle starts, internally you have to accept that that day onwards what starts is ovarian cycle also. in this cycle in this cycle, ovarian cycle, each ovary alternately in each ovary alternately means, you understand that if this month right ovary, next month left ovary will perform ovarian cycle. The other ovary shall remain silent with respect to the ovarian cycle in an alternate manner. So one ovary at a time, in which ovarian cycle will go on, and next month another ovary will experience such cycle. In this cycle, in each ovary alternately, one of these follicles, one follicle out of those 60 to 80,000, it will undergo maturation and development. You can imagine that there are 80,000 of them in each ovary. And out of those 80,000, just one follicle, primary follicle, gets a chance to undergo maturity and development in a given month. And that too in a one ovary at a time. That particular follicle will be stimulated by FSH, the follicular stimulating hormone, one of the gonadotropin secreted from anterior pituitary, shall target the ovary, and inside the ovary, its target shall be the primary follicle. How many primary follicles? One only at a time. In how many ovaries? Only one ovary. This follicle, under the stimulation of FSH, starts increasing the number of granulosa cells. So layers of granulosa cells increase. At that stage, you consider this as secondary follicle. This is called as secondary follicle. Now the secondary follicle will develop into next level follicle, which is called as tertiary follicle. Now these changes are very rapid. Within 6-7 days, 6 to 8 days, a primary follicle goes through secondary follicle stage and soon turns to tertiary follicle level. If you see the tertiary follicle, it has first of all some speciality. For example, it has got two coverings around itself, which are called theca, theca externa and theca interna. The outer one is called as theca externa and inner one is called as theca interna. So two such theca develop around the follicle. Also, there is an additional feature that you can make out over here, and that is there is a cavity inside the this tertiary follicle, which was not clearly apparent in this primary and secondary follicle. And this fluid filled cavity inside the tertiary follicle is antrum. It is termed as antrum. So tertiary follicle is characterized by presence of fluid filled cavity called as the antrum and the two layers around the follicle called as theca externa, the outer layer, theca interna, the inner layer. Of course, inside this, there is still that cell which is termed as, just a minute, what should we call this cell as? One which is surrounded by the granulosa cells and antrum cavity. What should we call this cell in the tertiary follicle as? It should be termed as what it is termed as? Primary oocyte. Nidhi Raval, absolutely wrong. Primary oocyte. See, in a primary follicle or in a secondary follicle or in a tertiary follicle, each one is an ovarian follicle wherein primary oocyte is surrounded by granulosa cells. Just the number of layers of granulosa cells are different, least in primary, more in secondary, and highest in tertiary. One more point, that the tertiary follicle has a specialized fluid filled cavity called as antrum, which was not so prominent in primary and secondary follicle. The coverings around the follicle are very prominent in tertiary follicle, that is outer one theca externa and inner one theca interna. Now the tertiary follicle has to grow further into what is called as Graafian follicle. Now this is the last stage in the follicle maturity. Um, this transformation of tertiary follicle to the Graafian follicle, what is the difference? One, size of the antrum is increased a little bit. Rest of the things are same. But there is one major event that is happening in between. And the name of that event is completion of meiosis 1. You remember this primary oocyte had just started the meiosis and got arrested in its prophase 1 when it was in ovary, fetal ovary. And since the birth till puberty, it has not just continued that suspended, that arrested meiosis. It had been blocked over there, stalled over there. That stalled or blocked meiosis will now proceed, and this cell will give rise to two different cells. This diploid cell will give rise to one small haploid cell called as first polar body, with a very little cytoplasm around the haploid nucleus. Another cell is called as secondary oocyte, the haploid cell over there. So within the Graafian follicle, you can see that there is a secondary oocyte, which is a haploid cell retaining large amount of the cytoplasm of primary oocyte at the time of meiosis 1, when the two cells were, daughter cells were separated. And other daughter cell was just a haploid nucleus and a very little amount of cytoplasm. It is also an haploid cell, a first polar body. And this is something which is transferring the tertiary follicle into what is called as Graafian follicle. It is termed as Graafian follicle. Have a look at that process again. I said that. Now, this is the meiosis, this is the point where the primary oocyte had gone into its meiosis and got arrested. Now, this is the primary oocyte which is found in the tertiary follicle. It now goes for completion of this meiosis. Formation of two unequal sized cells. Both are haploid, but the one which is with haploid nucleus and retaining the bulk of cytoplasm from the primary oocyte, it is termed as secondary oocyte. The other haploid nucleus with a negligible amount of cytoplasm is termed as first polar bond. And both of them surrounded by the granulosa cells inside the Graafian follicle. There you can see that. I'll just show you this Graafian follicle. Have a look at it. This is the secondary oocyte with its nucleus, cytoplasm, and it's a cell membrane. This cell membrane is further surrounded by now one more layer, and that layer is called as zona pellucida. This is termed as zona pellucida. That membrane of the membrane of the secondary oocyte itself is called as vitelline membrane. That is termed as vitelline membrane. Between the vitelline membrane and this zona pellucida, a glycoprotein layer, non-cellular glycoprotein layer, there is a first polar body present over there. You can see that. Can you see that? Between the vitelline membrane, the plasma membrane of secondary oocyte, and the outermost glycoprotein layer called zona pellucida in between these two. Actually, the space in between these two is called as perivitallin space, that is termed as perivitallin space. So in that perivitallin space, first polar body shall be found over there. As you can see, this is the antrum, the fluid filled cavity. These are the cells called as granulosa cells. These are the granulosa cells over there. These are those two layers, theca externa and theca interna. Outer one is theca externa, inner one is theca interna. So this is how the Graafian follicle appears to be. Towards the 14th day of the cycle, I told you what is the 14th day. You can understand that in the ovary of a female, which is the 14th day, based on her menstrual cycle. The day she had begin the first day of menstrual cycle with the menstruation starting over there as a first day of menstrual cycle. It is also the first day of ovarian cycle. So since the first day of menstruation, the 14th day, if you consider, so that is the 14th day of ovarian cycle also. Initially, in first few days, it was the FSH which was dominant one. Towards the 14th day, it is the LH. I hope you understand the full form of LH and source. You remember that one? It's the LH stands for what, by the way? Yes, LH stands for what? Which hormone? Luteinizing hormone. It stands for the luteinizing hormone over there. These luteinizing hormones surge. Surge means high level towards the 13th and 14th day of the Graafian follicle, forces the Graafian follicle to rupture itself and release the secondary oocyte. This is the secondary oocyte from the Graafian follicle coming out of it. Rest of the mass, the remaining granulosa cells and layers around the Graafian follicle shall be left behind in the ovary. And what is released that has to go into that, uh, obviously into that fimbriae, through the fimbriae into the oviducal funnel, fallopian tube in which it has to infundibulum region. There it is supposed to enter this secondary oocyte. This rupturation of Graafian follicle and release of secondary oocyte into the body fluids, it is called as ovulation. It is termed as ovulation. Now, oogenesis is incomplete. If you are smart enough to understand that, we have just reached up to this point, not gone forward, isn't it? So we are reaching to the stage where oogenesis has gone up the point where secondary oocyte is there along with the first polar body and it is liberated from the Graafian follicle. One such secondary oocyte shall be liberated into the body fluids in a given month on 14th day. एक female में 14th day of the cycle, obviously you know which cycle I'm talking about, is the day when she ovulates because of the 12th and 13th day LH surge, very high level of LH on 13th day. It is responsible for rupturation of this Graafian follicle and release of the secondary oocyte into the body fluid. And from the body fluid, it will enter into the infundibulum region of the fallopian tube, and that is facilitated by the fimbriae. I will be sending one video to show this particular event precisely. Now I will show you the structure of the secondary oocyte that arrives into the, or rather the one which is liberated at the time of ovulation, the one which arrives into the infundibulum region. It looks like this. Now I shall point out certain structures. You let me know the name of this one. The entire this, I'm not able to make it out. Yes, this cell. What you call this cell as? This is which cell? Come on, tell me. This is which cell? You are looking at which cell? What has happened to you guys? Reply. This is something which has been discharged from the Graafian follicle. Rest of the content was left behind. Theca, interna, externa, fluid, and all that stuff. Large amount of granulosa cell is left behind. This is our secondary oocyte. Is it gonna be haploid or a diploid cell? Yeah, it's an haploid cell. What about this structure in it? This is what for this secondary oocyte. This part represents its what? What is been represented over here? Tell me. Come on, this is the nucleus. What, you not able to make it out? What about this part of it? This is its ooplasm, the cytoplasm of secondary oocyte. What about this membrane of the secondary oocyte? Tell me. The whole cell is secondary oocyte. But what about this membrane of the secondary oocyte? What it is supposed to be called as? Tell me. Come on, you got to give a response. Vitelline membrane. It's called vitelline membrane. And together whole stuff is called secondary oocyte. Then what do you call this layer as? Tell me, what's the name of this layer now? Zona pellucida, isn't it? A non-cellular glycoprotein layer secreted by the granulosa cells around the secondary oocyte to keep it distinct. What you call this small structure, haploid structure between the zona pellucida and the vitelline membrane? What is the name of that structure? Yes, that is supposed to be first polar body. What do you call this space as? Yeah, this space is known as which space? Yes, it's termed perivitally space. It is termed as perivitally space over there. A space between oocyte cell membrane called vitelline membrane and zona pellucida is termed as perivitally space over there. Along with the secondary oocyte that is getting released into the released from the ovary. The secondary oocyte that is getting released from the Graafian follicle of the ovary. When majority of the mass of the Graafian follicle is retained back, you can see that some cells are at least released along with the secondary oocyte. You can see the same cell mass over here. So this few layers of the cells of granulosa category, granulosa cells, around the secondary oocyte, particularly outer to the zona pellucida. What you call this layer as exactly that is termed as corona radiata. It is termed as corona radiata. The outer layer of granulosa cells around this secondary oocyte, which is released after the ovulation, that secondary oocyte remains surrounded by this layer called as corona radiata. That is termed as corona radiata. Now, at this stage, the oogenesis is not yet over. Still, something is left. And what it is, that is something we will see in the next lecture. Madam, you can stop the recording. Now, if I ask you certain questions, then what is present in the primary follicle? In the center, what is present? Diploid primary oocyte. And in the surrounding, what is present? Oogonial cells, which is called as granulosa cells. Few layers of granulosa cells. That is the primary follicle. How about the secondary follicle? What change? No major change. Again, there is a cell called as primary oocyte, the diploid cell with arrested in prophase 1 of its meiosis 1, and rest again few more layers of granulosa cells. How about the tertiary follicle? Well, the cell is the same, primary oocyte, a haploid cell arrested in its prophase 1 of meiosis 1. But surrounding that, few more layers of granulosa cells. One large cavity filled with the fluid called as an antrum. And the whole follicle is surrounded by two extra layers, theca externa and theca interna. Again, what is there in the Graafian follicle? Now, this transformation is different. Primary, secondary, and tertiary follicle have a primary oocyte, but Graafian follicle possesses secondary oocyte. So, what happened in between? What happened in between this transaction, this transformation? So, during this transformation of tertiary follicle into the Graafian follicle, the primary oocyte of the tertiary follicle had undergone completion of meiosis 1. So from primary oocyte, one large cell which has retained large or bulk of the cytoplasm from the primary oocyte and got one haploid nucleus from it. It is called as, this cell is called as secondary oocyte. The other one with a haploid nucleus but a very little cytoplasm, almost negligible one around it, it is called as first polar body. So in Graafian follicle, in the corner, there is one secondary oocyte, the haploid cell, and there is a small haploid first polar body presence over there. What other changes? So few more layers of granulosa, so size is larger in the Graafian follicle. What more? So then the antrum size increases, the fluid filled cavity, its size increases. Theca externa and theca interna are already there. So that is termed as Graafian follicle. Acha, the hormone which is now stimulating the Graafian follicle to force it to rupture and release this secondary oocyte along with the polar body, that is termed as process, that hormone is called as LH hormone, luteinizing hormone, and it is responsible for this ovulation act, the act in which the mature Graafian follicle bursts open to release the secondary oocyte into the body fluid. Obviously, from here it will go into the infundibulum part. With the help of fimbriae, it will be managed. The secondary oocyte that is been released, you see it's a cell which has haploid nucleus, its cytoplasm called ooplasm, it's a cell membrane called oocyte cell membrane, or you may call it as the vitelline membrane, and further outer layer of glycoprotein stuff which is called as zona pellucida, and the space between the zona pellucida and the vitelline membrane called as perivitelline space, with the presence of first polar body in that region, because it was pushed out from the oocyte and it was kept out of the oocyte but still close to it. So that is the first polar body. And these few cell layers around the secondary oocyte, they constitute what is called as corona radiata. So that is termed as corona radiata. Okay, ma'am, over.